Hello everyone, so today let's talk about nucleic acids. I remember that when I was learning nucleic acids, there was so much vocabulary thrown at me that I was like trying just to memorize every single little thing and not actually understand how the things are pieced together. Like a puzzle, they all fit together. So the way I want to approach this video is to break down and to show you how these things are pieced together, and hopefully that's going to help you memorize this topic much easier. Okay, so let's talk about it. So when you hear about nucleic acids, the two main types of nucleic acids are DNA and RNA. Okay, but now let's take a step back and look at nucleic acids in general. Well, nucleic acids are made of something called nucleotides. And now you're going to be like, what are nucleotides? So a nucleotide is a nucleoside linked to a phosphate group. Okay, I'm going to be nice and give you a simple definition. So for a nucleotide, you have a phosphate, a sugar, and nitrogenous base. And again, the nucleoside, as you're going to see, is going to be the sugar and the nitrogenous base. So again, you have a phosphate and your nucleoside, which the nucleoside, again, is made of a nitrogenous base and a 5-carbon sugar. So let's talk about sugar. So for sugar, it's a very complex topic, and you're going to see that in biochemistry. You'll learn about all, like different types of sugars like mannose and ribose and glucose and fructose and etc. But for this video, what I need you to focus on is these two sugars that I have on the screen. Now, before I get into the difference, a vocab that I want to throw out is that these are an aldopentose, and this is mainly biochemistry, or for my MCAT peeps, you have an aldose plus a pentose is going to be an aldopentose. Again, ribose and deoxyribose are 5-carbon sugars, and again, aldose from aldehyde, pentose 5-carbon sugar. And so again, you have an aldehyde with a 5-carbon sugar. So again, aldehyde is more organic chemistry. But to simplify, if none of that made sense, to simplify, look at the green circle. On the left, you have a hydrogen. On the right, you have an oxygen and a hydrogen. So the one on the left is lacking an oxygen. So that's deoxyribose. The one on the right has the oxygen. So it is ribose. Again, another way you can look at it is it's ribose without the oxygen for deoxyribose. Again, deoxy means without oxygen. So it's lacking an oxygen. So it's the same structure as ribose, it's just lacking that oxygen. Now, why is this important? Why does this matter? Well, if you think about it, if you go to the name DNA, it's called deoxyribonucleic acid. You might be like, okay, who cares? But the thing is, deoxyribonucleic acid is referring to the sugar deoxyribose. So DNA has deoxyribose, and RNA, on the other hand, is ribonucleic acid, which again is referring to the sugar ribose. So again, DNA is deoxyribose, RNA is ribose, meaning RNA contains the ribose as the sugar, DNA contains deoxyribose as its sugar. Cool. So not that bad. So what did I say in the beginning? Nucleotides are made of a phosphate, a sugar, and nitrogenous base. So what are nitrogenous bases? So nitrogenous bases, you can also hear them as nucleobases. It means the same thing. This can be further categorized into purines and pyrimidines. So let's talk first about purines. So there are there are many ways that you can learn this, obviously. The way I learned it is GAP, but I also hear a lot pure as gold. And what does that even mean? Okay, pure for purine, A is going to be for adenine, and G is going to be for guanine. GAP, again, guanine, adenine, and there are purines, right? Now, other than purines, you can have pyrimidines. And again, purines and pyrimidines are categorizations for nitrogenous bases. Nitrogenous bases, again, can be made of purines or pyrimidines, and guanine and adenine are purines. And for pyrimidines, just remember cute pyramids. So cytosine, uracil, and thymine. Again, Guanine, adenine, cytosine, thymine, and uracil, all of those are nitrogenous bases. But again, they are divided into purines and pyrimidines. So for the purines, remember gap, and for the pyrimidines, just remember cute pyramids, because it sounds the same as pyrimidine. I mean, it sounds very similar, is what I'm trying to say. So again, hopefully that will help you remember that. Another thing that I have that I don't have on the screen, a way to remember for the people that are taking the MCAT, how many rings. Look at the number of ends. So guanine has 2 ends, so it has 2 rings. Adenine has 2 ends, it has 2 rings. Cytosine and thymine have 1 end, so 1 ring. And just remember that uracil is the replacement of thymine and RNA, so again, only 1 ring. Furthermore, something else that you need to have a huge understanding of, like you really need to know and stress and emphasize, is that thymine is in DNA but not in RNA. Uracil is in RNA but not in DNA. So I'll emphasize that in the next slide. So main differences for DNA and RNA. For DNA, you need to know that it's double-stranded. Like, that's a huge difference, okay? Again, it has deoxyribose, not ribose, and again, it has thymine, not uracil. So when you're talking about pairing, you remember A goes with T and C goes with G, adenine with thymine, and cytosine with guanine. And again, the thymine is in DNA, not in RNA. RNA, on the other hand, is single-stranded. Before I go to that, another thing that I want to help you remember is, because some of you guys are doing the MCAT, and so some of you guys asked me the structure. How do I remember the structures? So thymine, it begins with a T, so that stands for 2 oxygens, so it has 2 oxygens, and thymine has an M in it, and the other ones don't have. So M stands for methyl, and again, methyl is organic chemistry, and that's the CH3, and that you can see right here. So that's the methyl group. Now, let's move on to RNA. What is something you need to know about RNA? Again, it's single-stranded. That's a huge main difference. DNA is double, RNA is single. Again, RNA is going to have ribose and it's going to have uracil. So how is the pairing going to be? Again, it's going to be different. Why? Because A is going to pair with U and C is going to pair with G, adenine with uracil, and cytosine with guanine. So again, you can see that you have A, C, and G in both DNA and RNA, but thymine is going to be in DNA and uracil is going to be in RNA. Now, for uracil, how are you going to remember uracil? Remember that it's thymine without the methyl group. So you remove the methyl, or the CH3, it's the same structure without that methyl group. So again, thymine has the methyl group. Here you don't have a methyl group. So that's how you can remember the structure. I hope you enjoyed this video. Please follow, like, share. Remember, it takes time to create the content, so please like and share, and thanks again for watching.